Hi everyone, Miss Stafford here. Today I'm going to be walking you through an example protein synthesis practice problem. So you have an assignment this week where you're going to be asked to take 3 protein synthesis problems and complete them. This is only after you've gone through the videos associated with this week. So you're going to be turning a DNA sequence, which will be found in the nucleus, into an mRNA or RNA sequence, which can go from the nucleus to the cytoplasm, into a protein sequence, which is built out of amino acids. So for your practice problems, you're going to see a given DNA sequence that I've already broken down into codons using these slashes. Remember that a codon is 3 nucleotides at a time that are read as 1 when you are turning RNA into amino acids. So the first thing you're going to want to do is make sure that you go ahead and make a copy of the actual assignment that you are going to be working on, because just like last week, it is a Google Doc that you'll have to make a copy of before you're able to do it yourself. So once you've done that and you're in your own assignment, okay, you're going to see something like this, except you have 3 of them and your codon chart is over on the side. So the first thing we need to do is turn DNA into RNA. So RNA is also made out of nucleotides, and it follows the same complementary base pairing rules, except for the fact that there's no T or no thymine in RNA. Instead of thymine, as you saw in the video, it's going to be uracil. So now C still only complementary pairs with G, but A now pairs with U for uracil. So every place we would have put a T, we're now going to put an A. So the way this is going to work on your problems is you're going to come in here. You're going to delete the 3 lines. You're going to put your caps lock on and you're going to replace. So we see AAT. So now I'm going to remember my base pairing rules of A to U now instead of A to T, and C with G. So every place I see an A, I'm going to type a U. Caps lock on. Type a U. Now I see a T, so T goes with A. If you need to, take a second and put a little cheat sheet somewhere down here on the side. A equals U, T equals A, C equals G, G equals C. Okay, so now I'm going to go on to my second one, delete, 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 and see T must go with A, G with C, C with G. And now I'm going to continue across, filling in each of my codons. As I go, I'm just double checking that I only have 3 nucleotides in each one. Okay, I'm making sure that I'm filling out the correct one. If you're not sure, you could highlight the one that you're on to make sure that you can keep track of where you are. And it'll take a minute, but it's not too hard to do this part. You want to make sure that you type them correctly, because if you remember back to what we talked about last time we were face to face, if I make a mistake while I'm doing this, or if a cell made a mistake when it was doing this, it would be the equivalent of a mutation, which we don't want. Okay, now let me see if I've got this all right. Let's see if we have the right amount. All right, A to U, T to A. T to A, G to C, C to G. G to C, C to G, T to A. G to C, A to U, C to G. C to G, A to U, A to U. G to C, T to A to U. A to U, T to A, T to A. G to C, A to U. C to G, C to G, C to G. A to U, A to U, A to U. A to U, G to C, T to A, T to A to U, C to G. The reason they don't line up perfectly is just because in this font, letter G is wider than any of the other letters, so it's okay that it doesn't literally line up perfectly. All right, so we've made our complementarily bonded RNA. And that RNA would leave the nucleus and it would go to the cytoplasm where it would meet up with a ribosome. And now we need to translate from RNA or nucleotide language to protein or amino acid language. And to translate, just like you do from one language to another, we are going to need basically a language translator, but instead of from like English to Spanish, this is from nucleotide language to amino acid language. So we have that chart here. The way this works is we're going to do it one codon at a time. We see the nucleotide or codon letters in the middle, and then we have the amino acid names and the amino acid abbreviations on the outside. So let's see how this works. Our very first RNA codon is UUA. So you start from the center on yellow, and you find the first letter, U. You then work your way from the center out. So our second letter was U, so we're going to go from yellow to orange, U, U. And then our third letter in the codon is A, so we're going to go to red, U, U, A. That leads us to leucine, which I see is abbreviated LEU. So I'm going to type in LEU for the protein. Now for my next one, the codon is ACG. So I'm going to start from the middle and work my way out. A, C, G, threonine, abbreviated THR. Okay. Now I've got CGA. Start in the middle. CGA, arginine, ARG. You're going to keep doing this until you've made your way through all of the RNA nucleotides. So now I'm on CUG, CUG, leucine. Now you notice we had leucine already. So there are more than one codon for each amino acid. So there are only 20 amino acids, or 20 of these blue things around the side, but there are 64 possible combinations of nucleotides. So it's very possible like this, that even though you have different nucleotide sequences, different RNA sequences, you're going to get the same amino acid. So let's see where I am. I'm on the fifth one. 1, 2, 3, 4, 5, GUU. Start in the middle, GUU. Work my way out, and we've got valine. Then CAU. Start in the middle, work my way out. CAU, histidine, HIS. Now we've got UAA, UAA. Now this is very interesting. Notice it says stop. So I'm going to type that in, and then I'm going to highlight all of this and delete it. Because stop literally means stop. You've reached the end of the protein sequence, and that is the point when the protein would be completed, and that is the end of making that protein. So even though there were other RNA sequences after that, that is the point where I stop. So you guys are going to be doing this 3 times on your worksheet, which is on this week's assignment. I've given you the DNA. You're going to transcribe it into RNA using the A to U, T to A, C to G, G to C complementary base pairs. And then you're going to translate it into protein using this codon wheel right here, which will translate from nucleotide language, the yellow, orange, red, into amino acid language, the blue, with the abbreviation in green. Okay, email your teacher if you have any questions.